The duplication or replication of DNA begins with a single double helix strand of DNA, which is made up of 4 different nitrogenous bases. These are adenine, cytosine, thymine, and guanine. The bases are paired with a sugar phosphate backbone, which makes them a nucleotide. Adenine and thymine are always paired with each other, as are cytosine and guanine.
The DNA has a specific sequence on it at some point which looks like this. AT, AT, T, T, T. This is also known as the Tata box. And this is where the RNA polymer phase jumps onto the DNA to begin the process. Once the RNA polymer phase is on the DNA, it unzips the double helix and reads the single strand of DNA and calls up the appropriate matching nucleotide to build a strand of mRNA. However, the mRNA language is slightly different in that thymine is replaced with a closely related base known as uracil. The RNA polymer phase also rezips the DNA strand back together behind it as it moves downstream. Once the RNA polymer phase reaches the termination signal, it pulls off the DNA, leaving the mRNA behind as a new separate entity. Now, going back to the mRNA, it has a little bit of editing done to it, which finishes it off before it can leave the nucleus. First, a special type of guanine is added to the 5' end, known as the 5' cap. And then it has about 250 adenine nitrogenous bases added to the other end. This is known as the poly A tail. Once the mRNA has these things added, it is now free to leave the nucleus. Hooray!
This is where DNA replication and transcription are different. In transcription, mRNA is formed, whereas in replication you are just creating multiple copies of the same DNA. There are 2 ways in which DNA is replicated. The first is just simple replication, where the DNA is copied in the same manner as transcription and a whole new DNA is built up. Or there is semi-conservative replication, where the DNA is split into 2 parent DNA strands, and are separated during cell division. Then the DNA halves are paired with a new daughter half, keeping 1/2 of the original parent DNA and 1/2 of the daughter DNA. Now the process of translation begins. First, let's talk about what a ribosome is. A ribosome is a small machine more or less that's built up of rRNA, enzymes, and other proteins. These are used for all sorts of tasks, such as reading mRNA and building new proteins. In translation, first the mRNA finds a ribosome and enters into it. The ribosome then scans the mRNA, reading 3 nucleotides at a time. This is called a codon. The ribosome then calls in the appropriate matching tRNA, which holds 2 things. The first being the anticodon, which is the matching pair to the codon on the mRNA. And on the other end of the tRNA, it brings with it an amino acid. As the tRNAs come into and leave the ribosome, they leave the amino acids behind, creating a polypeptide chain.
A quick side note. Amino acids are essentially built up of the same bases that are in DNA, just with uracil instead of thymine. Now that we have a polypeptide chain forming, these can get quite long, often several hundred amino acids long. Once the chain is long enough, you can see the primary structure forming a new protein. Depending on the environment, the amino acids can actually change. Sometimes, 2 amino acids will get a little touchy and start forming hydrogen bonds on the side, thus changing the shape of the polypeptide chain. Often, these chains are found in new structures, which look like a helix or a plated sheet. This is known as the secondary structure.
Once the chain is fully complete, it may fold in on itself to make a 3D structure. This is known as the tertiary structure. Also, this is now a fully finished protein. Some polypeptide chains may come together and bond to make one jumbo protein. This is called the Quaternary structure.
And that's all you need to know about DNA replication, transcription, translation, and so on. Basically, you know how DNA makes proteins. Yay!